This begins the lecture on Chapter 10, Receptors for Peptide Hormones, Neuropeptides, and Neurotransmitters. In this chapter, we'll consider primarily the water-soluble extracellular chemical messengers. In Chapter 9, we considered the lipid-soluble steroid receptors. First, we start with an image of the structure of the cellular membrane to remind ourselves that it's a phospholipid bilayer with the polar heads that are partially hydrophilic and the hydrophobic tails. So the outside of the cell and the inside of the cell are aqueous media, and the polar heads can tolerate that. And of course, this is the nonpolar region, so water-soluble molecules cannot push through the membrane. They have to bind to some type of protein through the membrane. And we'll take a look at some more detailed illustrations of how these proteins are structured. They obviously are not simple rectangles. First, we remind ourselves that there are many receptor subtypes or multiple receptor forms for nearly all the receptor types that we would be considering. For example, in this table we're seeing norepinephrine and epinephrine, the receptor classifications on the right. So there's the alpha and the beta. There's both the type 1 and type 2 for each of these, alpha 1 and alpha 2, beta 1 and beta 2 adrenergic receptors. For dopamine, for example, they're showing D1 and D2. This is more extensive than this now. And for serotonin, 5-HT1 and 5-HT2. There are subclassifications like the 5-HT1A and 1D. So it's more complex than this, but we're trying to keep it relatively simple. Acetylcholinergic receptors, we've already talked about the muscarinic receptor and the nicotinic receptor. This is metabotropic. This is ionotropic, as we know. Opioid receptors, there's mu, sigma, kappa, delta, and epsilon. We've seen some of these involved in integration for the opioid effects. Remember, opiate is exogenous, opioid is endogenous. The opioid effects on neuroendocrine interactions in the hypothalamus. Histamine, H1, H2, and H3. PGE, we're not too worried about that. Progesterone, the A and the B, we had seen that in our discussion of the progesterone receptor. And estrogen receptors, the alpha and the beta receptors, as we had seen in the prior discussion as well. So our focus now is more so on the more classical transmitters and neuropeptides and some of their receptor subtypes, although we won't focus on this in a great detail because this is not a pharmacology course. We will astonish ourselves a bit in looking at the serotonin receptors. There are 7 families and 55 sequences. This was a publication from the 1990s. I don't think this list has been lengthened much. But what it tells us is that there's a tremendous diversity in much that we don't know versus what we do know. So a long list of serotonin receptors, it's really just astonishing. So we'll try to keep it on a more generic level to appreciate the overall mechanisms of actions and the structural complexities for distinguishing them, such as the 3 distinct superfamilies of the membrane receptors based upon their function. There's the ligand-gated ion channel receptors. Remember, we talked about voltage-gated ion channels. Now we're talking about ligand-dependent gating for the ion channels. And they obviously do act as ion channels. For example, the sodium channel for the nicotinic cholinergic receptor. Sodium channel also for the glutamate non-NMDA, also called the AMPA or AMPA receptors. So we'll see a few others like this. Transmembrane regulated enzyme receptors act as kinase enzymes, and guanine nucleotide binding protein or G-protein coupled receptors that'll be the primary focus of our discussion. We saw, for example, the tyrosine kinase receptor as one of the types shown to interact with the steroid receptors in lecture in Chapter 9 on the progesterone receptor. And we'll talk a little bit about this, but primarily focus on the G-coupled receptors. They are a family of proteins that have 7 transmembrane spanning segments. They are membrane receptors linked to G proteins. There are trimers binding guanylyl triphosphate. And they're kind of two subsets, but it's more diverse than this. But for example, there's the G sub S that is stimulatory and the G sub I that is inhibitory. Let's look at a few examples of this. This is an example over here on the right-hand side of the ligand-gated ion channel, probably nicotinic cholinergic because it requires two ligands to activate it, and it opens when ligated, and the sodium ions come in. This obviously is the metabotropic, the second type we saw here, transmembrane regulated enzyme receptors acting as kinase enzymes. So when ligated, it autophosphorylates on the tyrosine residue, so it's a tyrosine kinase in this example, consumes ATP to become phosphorylated, and then to activate tyrosine kinases that will go on to phosphorylate other proteins. This is the classical type of metabotropic receptor that we've seen, the receptor with the 7 transmembrane spanning segments, coupled to this trimer, alpha, beta, and gamma. And when ligated, there is the dissociation of the G proteins that become mobile and can bind to other molecules, the second messenger systems, and activate them. And we'll look at a couple of these enzymatic second messenger systems as we proceed. Here on the right-hand side, we're reminding ourselves, of course, that it is a dynamic interaction between the drug and receptor. We saw this with the steroid receptors, where the corticosterone mixed with the membrane fraction given time would produce the drug receptor complex form. And then if we added cold, we could dissociate off the ligated so it can dynamically go back and forth and be in the ligated and unligated form. The ligated form will bring about the response, which can be ion or metabotropic, and some of these metabotropics not only affecting enzymes but targeting other ion channels, as we recall. So here on the left-hand side, we are taking a look at the nicotinic cholinergic receptor, and we see that it is a bit different. There's 1, 2, 3, 4 transmembrane spanning segments for this subunit, and then there are 5 subunits with these transmembrane spanning segments that are associated together in a complex in the membrane. There are 2 binding sites. We see that there's an alpha subunit shown here and a beta subunit shown here. We won't get into the complexities of this, but there can be different subsets of these receptors based upon the subunits that are associated together. Here we see a cross-section of 5 assembled subunits, 2 alphas and 3 betas of the nicotinic cholinergic receptor. We're just seeing alpha and beta subunits shown here, with the binding sites for the neurotransmitters shown, and obviously both of these have to be ligated to open the channel. We see the gate closer to the cytoplasmic end of this channel. So the ligand binds extracellularly, and the channel opens closer to the intracellular portion, and then there are the transmembrane spanning components. So here we see this illustrated a bit differently for the transmembrane G-coupled protein receptors. The epidermal growth factor receptor is a monomeric form. We see the extracellular space, the transmembrane spanning segment, and the intracytoplasmic portion of these. So we'll discuss all of these examples of particular ones to emphasize these points as we proceed. Obviously, extracellular for ligation of the transmitter or the extracellular chemical messenger, and intracellular for mediating the metabotropic type of an effect. The insulin receptor we see here is a homodimeric receptor, and when ligated, homodimerizes and then is activated to produce its intracellular effect. The beta adrenergic receptor, 1, 2, 3, 4, 5, 6, 7, 7 transmembrane spanning segments. This is of that superfamily that we saw earlier. Extracellular loops involved with binding the ligand, intracellular loops involved with association of the G protein coupling mechanism. We can go on to look at further illustrations of these examples. In this case, tyrosine kinase, so a monomeric. There's a polypeptide hormone, a receptor tyrosine kinase ligating with that. Tyrosine kinase is activated by autophosphorylation at the tyrosine residue. Once it is active, then it can phosphorylate other molecules. In this case, we see a GRB2 and a SOS. Then the SOS can activate the RAS so that it will dissociate from GDP and bind GTP and become activated. RAF contributes to it. And there's an active RAF, and then ATP to ADP, so it phosphorylates the MAP kinase to become an active MAP kinase when it's phosphorylated, and then this can activate. It is the activated MAP kinase, and then consumption of 2 ATPs to 2 ADPs will phosphorylate MAP kinase. That becomes activated MAP kinase. You see the 2 residues, a threonine and a tyrosine, that are phosphorylated to activate it. And this is mitogen activated protein kinase, so that's what the MAP stands for. The active MAP kinase can translocate to the nucleus, and transcription factors will be phosphorylated, such as FOS, ATF2, Jun, and Mike. We've kind of mentioned most of these, not so much the ATF2, but the FOS and Jun. And these, once phosphorylated, dimerize, either homodimers or heterodimers, bind to their consensus sequences on target gene promoter regions, affect gene transcription, ultimately protein synthesis, and the target cell is affected through genomic mechanisms. There are obviously divergent pathways for these to go off and accomplish other things in the cell for response as well. Here we're focusing on the genomic effect. Well, now let's take a look at the other type of receptor with the 7 transmembrane superfamily with the 7 transmembrane spanning segments. This is the beta adrenergic receptor, and we see that the amino end is faced out into the extracellular matrix. So the amino terminal is extracellular. And the carboxylic acid, the carboxy terminus, is intracellular. We also see the 7 transmembrane spanning segments, and this creates loops. Loop 1, intracellular loop 1, intracellular loop 2, intracellular loop 3, extracellular loop 1, extracellular loop 2, and extracellular loop 3. So intracellular loop 1 links transmembrane spanning segment 1 and 2, extracellular 1, 2 and 3, 3 and 4, 4 and 5. So kind of a tricky thing for you to remember there. Later we'll study different properties of different loops. Intracellular or cytoplasmic loop 1, 2, 3 might have some special characteristic to accomplish some goal internally. And the extracellular tail or intracellular tail might have some functional properties associated with them. This is a bit more of a three-dimensional appearance of this. You see the amino terminus, carboxy terminus, and these 7 transmembrane spanning segments form this type of a complex of the transmembrane integral protein, in this case the beta adrenergic receptor. Here we're looking at another membrane receptor model. In this case, it is the general topography of the gonadotropin receptors, FSH and LH. And we see they also are in the superfamily of the 7 transmembrane spanning population, and they are Gcoupled. As well, we see the amino terminus in the extracellular domain, the transmembrane domain, the loops, the 7 loops. Obviously, these have lipophilic amino acid sequences, so they can tolerate staying within the membrane. We have intracellular loop 1, 2, and 3, extracellular loop 1, 2, and 3, and the cytoplasmic tail. So there are 3 domains: extracellular domain, the transmembrane domain, and the cytoplasmic domain. So that was one way of illustrating it. Here is another way of illustrating it, the actual amino acid sequences. And this is the deduced amino acid sequence of the LH chorionic gonadotropin receptor. The amino terminal half of the receptor contains 6 N-glycosylation sites. We see those in bold here. So the extracellular portion is glycosylated, so post-translational processing. The truncated receptor, representing extracellular domain N, is capable of binding hormone at multiple contact points with an affinity comparable to the intact receptor. So we see that there's 2 possible isoforms of this receptor, the full receptor and the truncated receptor. Truncated receptors usually are altered in their carboxy terminal end and quite often are involved in binding and transport of hormones without intracellular signaling activation. So you can see 341 residues in the extracellular domain at the amino terminal end. The transmembrane loops, remember there are 7 of them, 256 residues, and if we knew our biochemistry well enough, we should be able to look through here and see many of the lipophilic types of amino acid residues, whereas these are more hydrophilic as well as these. But remember, the transmembrane loops include both the transmembrane segments as well as the extracellular and intracellular loops. So we would rotate, that's not really the term I'm looking for, but we would switch from lipophilic to hydrophilic, and then back to lipophilic, and then hydrophilic, and then so on and so forth for our transmembrane spanning segments. And then, I don't know if these are the exact portions of them, I'm just pointing to parts that might be extracellular loop, and then transmembrane, then intracellular loop, and so on and so forth. I didn't do that right. It would be transmembrane, intracellular loop, transmembrane, extracellular loop, somewhere along here, and so on and so forth, until we got to the carboxy terminus, the cytoplasmic tail that would be contained within the cytoplasmic compartment. Here we see the amino acid sequence of the human pituitary GnRH receptor. As we proceed throughout our studies of the level of detail, we will focus on the expression of these receptors in their cells, the corresponding cells, and the gene expression regulation of them. But for now, we're just using them as examples. The GnRH receptor, 2 sites of potential N-glycosylation exist, one in the N-terminal extracellular domain and one in the first exoplasmic, the extracellular loop in bold, excuse me. So here we see the extracellular domain of the GnRH receptor, 35 residues, glycosylation at this location. Then the transmembrane loops, about 293 residues. To get those 7 transmembrane spanning segments, you need roughly 200 to 300 amino acids. And here's the carboxy terminal. What we do notice in these 2 is that we're showing an extracellular domain and transmembrane loops, but not an intracytoplasmic tail for the GnRH receptor. So it'll be interesting to study details on the GnRH receptor and how it functions. Here, for example, we see it again, the general topography of the gonadotropin receptors, as we saw from the previous slide, that had the amino terminal, the transmembrane spanning portion, and the cytoplasmic tail within the cytoplasm. So we wonder why we don't see all three domains illustrated for the GnRH receptor. We're just making generalizations at this point and not focusing on them specifically. We'll get into that later. What we have now is an appreciation for the diversity of the types of membrane receptors, either one transmembrane spanning segment or maybe four, and then you get the molecular complexing of the subunits, such as the nicotinic cholinergic receptors, 5 subunits together, each of which have 4 transmembrane spanning segments, and then the 7 transmembrane spanning segments superfamily utilizing the G protein mechanism for mediating the intracellular response. Now let's think about receptor regulation. So there can be homospecific and heterospecific, the two most common, and then there can be non-hormonal factors or genetically or programmed de novo or receptor synthesis that would be stage specific, such as developmental. So here we're thinking windows of expression of transcription factors that just bring about the gene expression for the receptor synthesis, de novo meaning of new, so brand new receptor synthesis, not being expressed at one point and developed, then suddenly being expressed, then maybe disappearing again or changing to a different level throughout throughout development. Excuse me. Non-hormonal factors, there can be viral agents or extracellular chemicals, for example sodium, that can impact the receptor regulation. Let's now consider the two most common types, the homospecific and the heterospecific. So obviously this is affecting themselves or affecting some other receptor, or being affected by some other receptor mechanism. Two ways of thinking about this. So first, for the homospecific, the hormones can regulate the number of their own receptors as well as the sensitization. So there can be desensitization and sensitization, supersensitization, as well as decreasing the numbers as in downregulation and upregulation. Heterospecific would be hormones that can regulate the receptors for other hormones or other transmitters. And we will have a set of research articles in a separate presentation that one of the papers will talk about heterospecific receptor regulation. So let's continue thinking about receptor plasticity either at the membrane or the genomic levels. So this would be sensitization, desensitization, and this would be downregulation and upregulation. So there's local regulation at the axonal or dendritic processes and terminals or the hormonal target cells, where you can increase activity or decrease activity. So increased activity at the receptors by an increased amount of the chemical messenger will cause desensitization mediated by kinase, so phosphorylation of the receptor, and in some cases even dephosphorylation. So phosphorylases activated that will alter the receptor and uncouple it essentially from the response mechanism, the second messenger system. Or downregulation through endocytosis and translocation into the cell, as well as decreased gene expression. Or if there's a decreased activity at the receptor, decreased amount of the extracellular chemical messenger, you can get supersensitivity. So the opposite of this, from a basal homeostatic level to an increased responsiveness to a small amount of the transmitter. As well, there can be upregulation to increase the synthesis. Remember, messenger RNA levels would go up, protein levels go up, insert it in the membrane, and then there's more available to be activated, by the low level of the chemical messenger extracellularly. Let's think about receptor internalization or receptor-mediated endocytosis of ligand-receptor complexes. In this case, it's not a neurotransmitter. Actually, it's lipoproteins that are binding to the receptor, but it shows the occupied receptors so nicely, I thought we'd use this to make this to illustrate this point. So here in this cartoon below, we can see the receptors are occupied. Somehow the receptors get sequestrated into an area where protein accumulates on the inside of the membrane, the cytoplasmic side. That protein is clathrin. So what is produced is an indentation on the surface plasma membrane facing the extracellular fluid, and then this becomes deepened into clathrin-coated pits. So the receptors and their ligand are being internalized until it is pinched off and forms an endocytotic vesicle. Typically, these that are clathrin-coated are targeted for the endolysosomal system, and these will be destroyed. So this is how you can get the endocytosis and destruction for receptor downregulation. Now there's other means for the endocytosis where it would not be clathrin-coated, and you would get an endocytotic vesicle, but it would not be clathrin-coated, so that vesicle can sit there and be available to be delivered back to the membrane to reinsert the receptors into the membrane. So this form of translocation can be two parts, translocated in clathrin-coated pits to the lysosomes to be destroyed for persistent downregulation. There would also be, through receptor-mediated mechanisms, a decreased gene expression and protein synthesis, so less protein, receptor protein available to be added to the membrane. Or it can just be temporary, translocated into a non-clathrin-coated pit, therefore not sent to the lysosomes, and sitting there and be available to be inserted back into the membrane when the extracellular chemical messenger concentration reaches basal levels and you want to get the normal level of receptors present on the extracellular membrane. So in studying membranes, membrane receptors, there are a variety of approaches. You homogenize the cells, you can collect just the cytoplasmic membrane, you can collect the vesicles, you can collect the cytoplasm without the membrane. You can thereby study membrane receptors on the surface, available to be delivered to the surface or on its way to destruction. And of course, those new ones being synthesized that are in the ER, and so on and so forth. So sophisticated methodological approaches allow you to differentiate these distinct populations of receptors. Let's look at the receptor internalization for downregulation here with the endocytosis of the ligand receptor complex in this illustrative cartoon. The ligand binds the receptor. The ligated receptors are sequestered to a clathrin-coated pit. You get the clathrin-coated endocytotic vesicles that end up with fusion to become endosomes. Then, because of the clathrin coating, they are sent to primary lysosomes, and they can become then secondary lysosomes upon fusing with those primary lysosomes, and you can get dissociation of the ligand as well as degradation of the receptors, or just degradation of the receptors, another theoretical model, so that you can get the receptor recycling and insertion back in to the membrane. So what I had described to you was two mechanisms. Either everything's the same up to the point where you either have clathrin-coated or non-clathrin-coated pits. Clathrin-coated target the lysosomal system. The non-clathrin-coated target just sitting around and waiting to be recycled back to the membrane. So divergent theories here, but we understand these mechanisms of downregulation. The excessive signaling of the receptors as well will target the nucleus somewhere down inside the cell, of course, to decrease the gene expression for this receptor population. So you decrease synthesis and decrease the number of available receptors on the surface of the cell, and that is the true downregulation. Desensitization can be uncoupling from the signaling mechanism and internalization as well. These are not available to be activated. So there's a diverse way of describing this desensitization downregulation. They are two distinct measurable events biologically and chemically, biochemically, but they are a continuum, and we have to think of them that way. First, you uncouple, desensitize, endocytose, and either hold them to be reinserted or send them through the lysosomal system to get rid of them permanently. Let's now begin to focus on receptor transduction, with focus on the guanine nucleotide binding proteins, so that 7 transmembrane spanning superfamily. So the signal transduction by the G proteins, you are referred to table 10.1 in the textbook, asking can you memorize it. We won't ask that level of extension, just a generalized appreciation of all the different G proteins and how they function, although we will look at a couple of tables. So there are 12 different types of G protein superfamilies. 4 stimulate second messenger synthesis, the G sub S, such as cyclic AMP synthesis. There's a WebCT link to see how this cyclic AMP system works in a cartoon type of illustration. 3 inhibit second messenger systems, the G sub I. And then there are others that regulate. So here's 7. So that means there's 5, 4 of which are shown here, that can regulate calcium channels, so a G sub naught, potassium channels, a G sub K, phospholipase A G sub PLA, and phospholipase C, a G sub PLC. We will focus on this G sub PLC system, as well as stimulators and inhibitors of the adenylate cyclase, cyclic AMP signaling mechanisms. So our focus is primarily on 2 second messenger systems, the cyclic AMP and the phospholipase C mediated IP3 system. So let's now transition over to a discussion of receptor transduction. And we consider a mechanism for converting receptor activation into neuronal function, the transmembrane signal transduction. So modulation of cyclic AMP and modulation of phospholipase C that generates inositol triphosphate, or IP3. And so it's the cyclic AMP and IP3 second messenger systems that we will discuss in detail. We've seen this before. ATP is used to generate the cyclic AMP and activates the cyclic AMP there. After activates protein kinase A, increasing protein kinase A activity. The modulation of the phospholipase C system generates inositol triphosphate and diacylglycerol, both of which are second messengers. The IP3 will release internal calcium, and diacylglycerol will stimulate protein kinase C. So protein kinase A, the A for cyclic AMP, protein kinase C, the C for phospholipase C. So you will have to recognize the distinction between these two protein kinases. So here we see the enzymatic formation and inactivation of cyclic AMP. Again, we remind ourselves of the life cycle of molecules. Magnesium acts as a bridging cofactor between ATP and adenylate cyclase, and the metabolite of cyclic AMP, the 5' adenosine monophosphate, is inactive, so we inactivate the second messenger. So we know that ATP is converted to cyclic AMP by adenylate cyclase, and it is 3', 5' cyclic AMP. So I don't know which is the 3 and the 5. 1, 2, 3, 4, 5. Let's go that way. 3' to 5' cyclization here. And then phosphodiesterase is the enzyme that will break this cyclization, so you have the phosphate ending up on the 5' carbon with 5' adenylate adenosine monophosphate. So if it is not cyclized between the 5' and the 3' excuse me, 3' and the 5' carbon, phosphate ends up here to produce the 5' adenosine monophosphate. Pardon me for the error there. If you don't have that cyclization between the 3' and the 5' carbons, then it cannot bind to its binding site for activation. So the cyclic AMP can have 3 primary actions. It can alter the permeability of the cell membrane. How? Well, we've seen that before. Figure 10.5 illustrates it. It alters enzyme activity by increasing the degree of phosphorylation. It activates protein kinase A, which goes on to phosphorylate and activate, and goes on and on and on. And it can stimulate protein synthesis, having genomic effects. We've already talked about how protein kinase A phosphorylates a transcription factor, CREB, C-R-E-B, cyclic AMP, response element binding protein. And that way we have the divergent effects by acting on enzymes and ion channels to impact the cellular responses either at the membrane through permeability or intracellular metabolic changes, and then acting on the transcription factor to get at the genomic effects for regulating transcription rates. So the genomic effects of the cyclic AMP, we get ligation of the receptor, the effect on the G proteins, the activation of the G sub S, GTP binds to the G sub S, and it activates the adenylate cyclase. We're not looking at the complexity of this in this simplistic model, obviously. And then at step 5, the ATP is converted to cyclic AMP. Step 6, the ATP binds to the regulatory or the inhibitory subunit of the soluble protein kinase, the cytoplasmic. And then at step 7, because of that ligation, you get the dissociation of the catalytic subunit from the regulatory subunit of the protein kinase, and then you get nuclear translocation of the catalytic subunits and phosphorylation of nuclear regulatory proteins, in other words, transcription factors, and you impact transcription. For cyclic AMP, of course, this is CREB that is in the nucleus acting as one of the nuclear regulatory proteins. So also there can be competitive stimulatory and inhibitory transducer G proteins in hormonal regulation of adenylate cyclase. So hormone S acts through G sub S to increase adenylate cyclase activity. Hormone I acts through G sub I to inhibit it. So the presence of these two antagonistic hormones in differing levels with activation of their corresponding receptors determines that at any one point in time what would be the level of adenylate cyclase activity and therefore the concentration of cytoplasmic cyclic AMP. Now let's take a look at the catalysis of the phosphatidyl inositol, the PIP2 biphosphate, so PIP2. This is one of the phosphatidyl, the phospholipids within the membrane. The phospholipase C is also in the membrane, and when it is activated through the Gcoupled mechanism, its substrate is right there in the membrane, and it changes it to IP3, inositol triphosphate. This diffuses into the cytoplasm, and diacylglycerol stays in the membrane and is mobile and will move around, and if it bumps into a target molecule, it can cause a cascading event. So let's take a look at that in this cartoon illustration. Here we have 1, 2, 3, 4, 5, 6, 7 transmembrane spanning segments. N-terminal extracellular domain tail, carboxy tail. You see that the carboxy terminal, or the carboxy tail, has a portion that interacts with the G proteins. Excuse me. And when it is ligated, it will, of course, alter the G protein. What would the G protein subclass be here? G sub PLC, and it activates the membrane protein phospholipase C. The PIP2 is a membrane phospholipid. Its substrate is right there. Produces the byproducts of diacylglycerol and IP3. The IP3 diffuses into the cytoplasm. The diacylglycerol can float around in the membrane, can activate the membrane form of protein kinase C, can act on nearby proteins to phosphorylate them and contribute to the cell response. Also can act on membrane ion channels and alter the permeability of the membrane. The IP3 binds to a specific receptor on endoplasmic reticulum, the endosomal system, that sequesters calcium. These calcium stores are then released as this ligand gated ion channel on the membrane binds the IP3, the cognate ligand, and calcium will diffuse through the channel into the cytoplasm. Intracellular free calcium levels contribute to the cellular response. And we'll talk about calcium being a second messenger system in a bit more detail as we proceed. So here we see the cartoon illustration of the phosphoinositide second messenger system. I think you're beginning to see the potential for using these cartoons on exams where you would have to fill in maybe not all of them, but certain of these key steps. So we see the hormone binding to the receptor. The receptor is ligated. We get activation of the G protein. It binds GTP and is activated, and then activates the phospholipase C. So this is the G sub PLC type of G protein. Increases phospholipase C activity, converts PIP2 into IP3, acting on the endoplasmic reticulum for the mobilization of the intracellular calcium. And now what we're building a story on is you can have intracellular free calcium concentrations contributed to by intracellular sources. If a calcium ion channel on the membrane is opened, then you can have extracellular contributions. So we'll look at some studies where they use various procedures to determine whether a cellular response is produced by extracellular or intracellular calcium. Excuse me. Number 8 here, we see the IP3 can also activate guanylate cyclase, converting GTP into cyclic GMP that can activate protein kinase G. So now we have the diacylglycerol activating protein kinase C, and as well the IP3 system, the phospholipase C second messenger system, can activate protein kinase G. So these divergent pathways of the phospholipase C system contribute at many levels to the alterations in the target cell function. Here we can take a look at it again, focusing on the intracellular calcium regulatory mechanisms. The source of this illustration is shown above. So we have the agonists such as hormones, growth factors, and other effectors acting on the G protein receptor or protein tyrosine kinase that can activate phospholipase C such that PIP3 produces the diacylglycerol in the IP3. The IP3 is degraded to metabolites of the IP3 to inactivate it, obviously. But while it is in the active form, it binds to the IP3 receptor on the endoplasmic reticulum, part of the endosomal system, the intracellular calcium stores. Excuse excuse me. And the calcium sequestering pool in there is at a higher concentration than in the cytoplasm. So when this channel opens, the calcium will move into the cytoplasm. It moves out much greater than it moves in, obviously. So we have to ask the question, how do we get calcium back out of the cytoplasm? Well, in part, this is brought about by the diacylglycerol acting on the PKC that they think or are hypothesizing that it will activate the SERC, the sarcoplasmic reticulum calcium channel, calcium pump, excuse me. Obviously studied most extensively in muscle tissue, therefore sarcoplasmic, but it would be the endoplasmic calcium pump that will actively pump the calcium back into the calcium stores. The calcium does leak out and in from extracellular sources as well, and so you have calcium homeostasis occurring within cells with all these dynamic mechanisms. So calcium is a ubiquitous second messenger, important in cell growth, differentiation, signaling, and so on and so forth. So we will spend a bit of time talking about calcium generally here as we proceed. But first, let's take a look at some of the cell surface receptors and their transducers. This is the group 1. So the receptor or hormone is shown here, and the major transducers. Beta 1, 2, there's a 3 now. G protein to adenylate cyclase. Alpha 1 adrenergic, phospholipase C beta, so different isoforms of phospholipase C. Alpha 2 adrenergic, PLC beta and cyclase. They decrease it, so this would be G sub I. This would be a G sub S. Muscarinic, the M1 type subtype of the muscarinic receptor, PLC beta. D2 dopaminergic, PLC beta, and G sub I for adenylate cyclase. Histamine, PLC beta, angiotensin, bradykinin, angiotensin, vasopressin, also acting on PLC beta. Glucagon on the cyclase, calcitonin on cyclase, parathyroid hormone on adenylate cyclase, and through the G sub I. Prostaglandin E2, this is G sub S, sorry about that. This is G sub I for inhibiting cyclase activity, and the leukotrienes acting on phospholipase C beta. Here's the group 2, the thromboxane, let's not worry about that. Thyrotropin releasing hormone, PLC beta. Thyroid stimulating hormone, cyclase. Follicle stimulating hormone, cyclase, and LH. We've seen these two already in an example in our discussions of the steroid hormone receptors when we were talking about the conversion of testosterone to estrogen. Epidermal growth factor through a receptor tyrosine kinase mechanism, also activating phospholipase C, but it's the gamma subtype. Another tyrosine kinase through platelet-derived growth factor, but PLC gamma. Insulin through IRS1. You'll have to forgive me, I don't know what IRS1 is. It's good that we don't all know everything, huh? Growth hormone, again a tyrosine kinase through a JAK-STAT system. Prolactin through a tyrosine kinase as well, the JAK-STAT system. And activin and inhibin, part of the TGF-beta family. But the inhibin will be very important when we talk about negative feedback, specifically on follicle-stimulating hormone. This is receptor serine threonine kinase through the SMAD system. So just an endless number of these types of things. So let's not get bogged down into it. Let's just remember the general G sub S, G sub I, PLC, and the G sub naught, of course, on the calcium channels. and the G sub K on potassium channels. So it reminds us of changing membrane permeability, either directly or through changing enzyme activity within the cell for divergent effects on permeability and metabolism. And let's not always forget about the genomic effects, because if we're stimulating secretion, we're also stimulating synthesis. So we keep that in mind. This molecule, this extracellular chemical messenger, acts on a membrane receptor to stimulate secretion and synthesis. That's our basic premise as we proceed throughout our discussion of endocrinology. So now let's focus a little bit on calcium, these receptor-mediated changes in intracellular calcium, and how calcium might be acting as a second messenger within cells. So the cytoplasmic level is usually very low, 10 to the minus 7 molar, so heptamolar, and maintained by a calcium pump. We saw that. If calcium increases, it can be either, this is intracellular free calcium concentration, right, to go above 10 to the minus 7 molar, either by opening the calcium membrane channels for extracellular calcium to contribute to that intracellular cytoplasmic rise, or by releasing from the intracellular stores within the endoplasmic reticulum or the endosomal system. The hormonal binding activates G-alpha subunit, a G sub Q, which is another way of talking about the phospholipase C, the G sub PLC system activating the phospholipase C, of course, bringing about the IP3 system. So what we are beginning to ask ourselves about is how a chemical or electrical signal in a neuron or other cells, maybe not so much electrical signal in cells that do not have excitable membranes, but a chemical signal that can have divergent pathways to cytoplasmic membrane ion calcium channels and signaling mechanisms to increase the calcium release from the intracellular stores, both contributing to, or either or, contributing to, or both, right? Let me say that correctly. Either or both contributing to the increase in cytosolic concentrations of calcium. Then it can bind to proteins such as calmodulin, altering enzyme activity, or other calcium binding proteins, altering exocytosis and intracellular movement or cellular movement in general. So there are multiple roles of intracellular calcium ions. Here we see the voltage-sensitive calcium channels, ligand-gated calcium channels, the PI-linked receptors for intracellular sources being shown here. However, there might be other calcium ion channels in the membrane that somehow, through the diverging pathway of the PLC system, are affected so that extracellular calcium can contribute as well to the IP3 mediated intracellular calcium released from the endoplasmic reticulum, so that you get an intracellular calcium concentration increase, can bind to calmodulin that can affect myosin light chain kinase for activation, for example, contraction in the smooth muscle through the myosin light chain alterations, or phosphorylase kinase to activate phosphorylase for dephosphorylating certain molecules, or CAM kinase, calcium calmodulin, kinase 3 for eukaryotic elongation factor 2. I don't know what that is. Or multifunctional CAM kinases that contribute to the cellular response. So the calcium has many divergent pathways for altering this, the target cell and bringing about a response in the target cell. So let's consider further the role of calcium as a second messenger, that calcium concentration in the compartment controlled by regulated activity of calcium transporters and channels located in membranes surrounding the cytosolic compartment. So the calcium concentration in the cytosol of the resting cell is maintained at very low levels, 10 to the minus 7 to 10 to the minus 8. The greater the negativity, the lower the concentration. Remember that. And remember, this is a 10-fold difference, power of 10. Greater than 10,000 times higher in the extracellular space or in certain organelles such as mitochondria, smooth endoplasmic reticulum. That's where the circ comes from too, the smooth endoplasmic reticulum calcium pump, rather than sarcoplasmic reticulum, as I had stated before. In the plant cell, the vacuole would be involved in this. So when it enters the cytoplasm, the calcium ions trigger a number of responses, as we had stated. So here's an illustration that can show some of the things that are happening. So you can have the cytoplasmic, the sensitive calcium channel, the voltage sensitive calcium channel. So when it opens, there'll be a calcium influx, and the IP3 receptor on the endoplasmic reticulum can open, and you can get the calcium efflux as well. I'm not exactly certain what this big red arrow is doing here, but anyway. So there might be a calcium channel inhibiting factor, where when you get the IP3 activated intracellular release, it might blunt the extracellular source as shown here. So it's estimated that calcium migrates no further than 0.1 to 0.5 micrometers, lasting only about 50 microseconds before encountering a binding protein, assuming 108 moles seconds per KON. Oh my God, what is that? And 300 micromolar binding protein concentration. So somebody did a lot of mathematics to just kind of figure out these types of things. Most importantly, what we realize is that there are calcium waves, but calcium doesn't travel very far. So if you release calcium here, it's not going to diffuse all the way across the cell. Somehow there's a general activation to get the calcium released to distribute it to different parts of the cell. And this was important when we talked about exocytosis, where the extracellular calcium was very important, produced a localized increase in calcium, not a general intracellular increase in calcium. So there was a localized effect of that calcium on the vesicular binding. So as would be expected from the number, affinity, and specificity of calcium binding proteins, calcium can be extremely localized as a second messenger, as we just implicated for exocytosis, but also for localized intracellular effects. The calcium diffuses much more slowly than predicted, simply from its ionic, about 1 angstrom diffusion coefficient, which equals 100 micrometers squared, 100 square micrometers per second, or even hydrated radius of about 800. So the local nature of the calcium around the release sites results in asymmetric distribution of the calcium-dependent proteins for specialized functions. The fine reticular endoplasmic reticulum spreads like a vast three-dimensional spider web within cells, acting as a framework for calcium-binding proteins and actively sequestering calcium into its intraorganellar space, which contains high concentrations of buffering proteins, for example, calsequestrin. So the point being made here is that you can get a more expansive release of calcium if somehow the membrane of the endoplasmic reticulum spreads this opening of the IP3, the calcium channels, and you have membrane ever present with the the transport protein to begin bringing calcium back into the endoplasmic reticulum when it is immediately available in the cytoplasm. So very short diffusion and very short time period that it is available to bind to the calcium binding proteins to bring about the second messenger function. So these calcium binding proteins first serve as a buffer or as a sensor to mediate the messenger role of the calcium. So some of the buffers in the cytosol are, for example, parvalbumin, calretinin, calbindin. We find these in many different neuronal populations. Buffers in the endoplasmic reticulum or sarcoplasmic reticulum, the calsequestrin, as we mentioned, calreticulin, a low affinity millimolar Km, the molarity at which you get 1/2 binding, high capacity 25 to 50 ions per molecule for calcium, and so a lot of calcium ions can be bound to a small number of proteins to sequester that calcium in the endoplasmic reticulum. The calcium binding protein of the endoplasmic reticulum and the BIP active in folding and the maturation of secretory proteins in the ER. So this calcium binding in the ER is not just for sequestration. It has divergent functions, especially related to post-translational processing of proteins, especially secretory proteins. There are a large group of proteins, more than about 30 subfamilies, which include the following: the parvalbumin and the calbindin. These are the buffers, because cytosolic free calcium is dangerous. That's why it's kept very low. It can damage cells and even kill cells. There are calcium sensors such as troponin C in the muscle that controls the interaction of actin and myosin in contraction, or calmodulin, such as we saw with the CAM kinases, and it is ubiquitous. So let's continue thinking about these divergent pathways and signal amplification for each of the pathways. So for one ligand that binds to a receptor, we can activate, in this example, 3 second messenger molecules that can go on each to activate 3 in this model, of course. So we've gone from 3 to 9, and now each of those going to 2. So one ligand is amplified into many intracellular molecules. So these can be enzymes or calcium, whatever it may be. There's an amplification so that the impact of one receptor being ligated on the cell is magnified. So we continue to consider this. The signal molecule on the membrane receptor, signal transduction by the effector protein, the amplifier enzymes, get the second messenger molecules acting on ion channels. So we have an amplification of the second messenger because we have an enzyme that's converting a lot of ATP to cyclic AMP. So we activate one enzyme, we get a lot of second messenger. Then that activates protein kinases that phosphorylate proteins and amplify the cell response. We also get the increased intracellular calcium, calcium binding proteins contributing to the cell response. Also, we remember that we amplify the signal by having inactive enzyme 1 being activated by the initial stimulus, being around to activate a lot of inactive B, being around to activate a lot of inactive C, acting on a lot of substrate to produce a lot of product. So this cascading amplifying effect can have persistent and dramatic effects on the target cell beyond, of course, just secretion and synthesis. For thinking of target cells where we impact the cell differently, not to release anything, then this amplifying effect is very important. For example, maybe activating carbohydrate metabolism enzymes changing the metabolic state of the target cell, or fat storage or fat breakdown, or the various intracellular mechanisms within neurons to upregulate and downregulate receptors. In addition, if we think of within neurons to have the spread of the signal so that an effect in the dendrites can contribute to response intracellularly at the level of the nucleus. So by amplifying the signal and having many different substrates spread and diffuse farther and farther, it's possible to have a receptor-mediated event in a dendrite of a neuron impacting somehow the nucleus. So these are some of the enigmas that we have yet to understand completely, how a dendritic second messenger neurotransmitter event can impact nuclear events, 10 microns away, 100 microns away. So these are some of the things that we still don't fully understand, but we recognize that they indeed truly can happen. Now let's just take a final moment here to talk about receptor binding. We'll go into greater detail on this later. There is an intrinsic activity, so we've talked about specificity and affinity. Affinity is mentioned again here. Now we add to that intrinsic activity. So affinity is the strength of the binding, as we said. So a kinetic value, K sub D, or KD, can be estimated. This is the dissociation constant. So if you have a high KD and a low affinity, you can have an effect that would be almost equivalent to a low KD, high affinity. So it would bind small amounts of the I'm sorry, let me back up. That still would be true, but this is really here to explain something else. A receptor with a low affinity and a high KD can still produce a significant response. But what it means is that when you have a high KD, the concentration at which you get 50% activation of the receptor population, or binding of 50% of the receptors, is higher. So this KD value is high if you have a low affinity receptor requiring a high concentration to activate it. If you have a high affinity, then a low concentration of the transmitter, I hope I was saying that here. Low affinity, you need high concentration of the chemical messenger, so you have a high KD. High affinity, a low concentration of the chemical messenger can bind to it at a good strength and activate it, so you get a low KD. A lower extracellular concentration of the chemical messenger will produce a 50% response, or 50% binding to the receptors. So if you see a receptor with a high KD, you naturally assume that it has a low affinity. If you see a receptor with a low KD, you assume that it has a high affinity. So for intrinsic activity then, an agonist has the same potency as the natural ligand, and an antagonist blocks the natural ligand. So a good agonist and antagonist should have intrinsic activity. Should have affinities that would, for the receptor, that would be similar to the endogenous ligand.